Good day, I'm Shivashni Silvanathan, and my presentation is entitled Genomics in Lung Cancer, What the Future Holds. Basic concepts in molecular biology is important because it provides a framework to how cells function. So understanding DNA structure, gene expression, and how molecular disruptions in these processes lead to disease is vital because the same information is used to identify targetable mutations for diagnosis and personalized treatment, especially in cancer. To start off, DNA or the deoxyribonucleic acid is the hereditary material in living organisms, and it's composed of two long strands that form a right-handed double helix. The DNA is housed within the nucleus of each cell. It's easy to visualize the DNA double helix as a ladder where the two side rails are made up of alternating sugar, deoxyribose, and phosphate group forming the sugar-phosphate backbone. The steps or the rungs of the ladder whereas are made up of paired nitrogenous bases, adenine, thymine, cytosine, or guanine. Adenine A pairs with thymine T via two hydrogen bonds and cytosine C pairs with guanine G via three hydrogen bonds. This twisted ladder is then compacted into chromosomes by tightly wrapping itself around proteins called histones. 8 histone proteins form a histone octamer around which the DNA strand is then wrapped for packaging into chromosomes. The central dogma of molecular biology explains the flow from DNA to RNA to functional protein. To put it simply, when a cell requires a protein for a specific function, the relevant gene is read through a process called transcription. The required protein, which is the workhorse of the cell for all functions from metabolism to immune response, is then made via a process called translation. To elaborate a bit more on both of these processes, transcription begins with RNA polymerase binding to the promoter region of the antisense strand, synthesizing a complementary RNA strand. This functions as you can say, a temporary photocopy of the gene sequence in exception that thymine now is replaced with uracil. The pre-mRNA is modified, whereby introns or non-coding regions are removed, exons or coding regions are spliced together, and a cap and tail is added for stability and export. This processed mRNA then leaves the nucleus and goes to a ribosome in the cytoplasm to begin the process called translation. During translation, ribosomes read the mRNA codons, 3 bases at a time, to assemble amino acids into proteins. tRNA or transferred RNAs bring the correct amino acids and catalyzes peptide bond formation. Protein folding follows to achieve functionality. In a very brief summary, that is how the genetic information stored in DNA is translated into proteins for cell function. So now we go into omics. So omics is the study of all the biological molecules which I mentioned earlier. For example, genomics is the study of the gene, proteomics studies the proteins. I would like to focus on cancer genomics, which identifies the genetic alterations that drive tumor development, progression, and treatment response. Here I would like to highlight some technologies used in cancer genomics. For the interest of time, I would elaborate on how it helps lung cancer specifically. So, NGS or next generation sequencing is the cornerstone of cancer genomic analysis. It enables high throughput sequencing at both whole genome and targeted levels. In clinical oncology, NGS is used to detect mutations, insertions or deletions, gene fusions, and copy number alterations. Panels targeting genes like EGFR, ALK, and ROS1 are routinely used for molecular profiling in non-small cell lung cancer. NGS is also essential for monitoring acquired resistance such as the EGFR T790M, which guide transitions to next-generation tyrosine kinase inhibitors like the osimertinib. Now we go on to liquid biopsy. This is a minimally invasive method that analyzes the circulating DNA in blood samples. This technique is particularly useful when tumor tissue is inaccessible or inadequate. NGS-based ctDNA testing allows for the detection of emerging resistance mutations such as, like I said, EGFR T790M, and MET amplification. And it also helps in assessment of minimal residual disease and real-time therapeutic monitoring. And it is now recommended by clinical guidelines for managing advanced non-small lung cancer. RNA sequencing whereas provides insights into the tumor transcriptome, enabling analysis of the gene expression, splicing variants, and fusion transcripts. It plays a vital role in identifying gene fusions such as ROS1 and ALK, and in assessing tumor immune microenvironment profiles. It also helps to differentiate responders from non-responders by highlighting gene expression signatures linked to TKI or drug sensitivity or resistance. Okay, this interpretation of this large amount of genomic data which we will get relies on powerful bioinformatic tools. There are many platforms for this, and these tools support oncologists in making evidence-based treatment decisions by translating complex genomic data into clinically actionable insights. Okay, so now moving on to my disease of interest, lung cancer. Now, lung cancer remains the leading cause of cancer-related mortality worldwide, so the magnitude of the disease is immense. It is broadly divided into two main categories: non-small cell lung cancer or NSCLC, which constitutes 85% of cases, and small cell lung cancer, which makes up the remaining 15%. Non-small cell lung cancer includes histological subtypes such as adenocarcinoma, squamous cell carcinoma, and large cell carcinoma. Traditionally, lung cancer diagnosis was simple. It was mainly made via imaging and a histopathological confirmation. But with the advent of molecular profiling, the diagnosis of lung cancer usually now involves its mutation and associated biomarkers as well. At the molecular level, non-small cell lung cancer is often driven by oncogenic mutations in genes such as EGFR, ALK, KRAS, ROS1, and BRAF. Emerging biomarkers including tumor mutational burden and microsatellite instability and also PD-L1 expression guides immunotherapy selection. These markers are often assessed using the same NGS platforms. Treatment strategies in lung cancer whereas depends mainly on disease stage, the molecular profile, and patient fitness. Localized cancer is usually treated with surgical resection followed by chemotherapy or radiotherapy, whereas advanced cancer is managed with systemic therapies. Patients with driver mutations benefit from targeted therapies such as the EGFR mutant tumors, which respond to tyrosine kinase inhibitors or TKI like gefitinib and osimertinib. Resistance mechanisms which develop in these patients, such as EGFR T790M mutation, necessitate a switch to newer generation TKI, meaning a patient has to be switched from first generation gefitinib to the third generation osimertinib because of resistance. Patients without actionable mutations may receive immune checkpoint inhibitors targeting PD-1 or PD-L1 in combinations or alone. Okay, so now we come to the term exceptional responder. So in oncology, exceptional responders are basically patients who exhibit complete or near complete remissions or longer progression-free survival than expected. These cases offer unique insights into tumor biology and is a promising focus in the field of multi-omics technology. I would like to share a case report of one of our patients in National Cancer Institute Putrajaya, who is an exceptional responder. So she's a 59-year-old female. She has underlying hypertension and chronic kidney disease stage 4. She presented to us in 2016 with persistent cough. Imaging and biopsy confirmed invasive adenocarcinoma, which is a type of non-small cell lung cancer, and it harbored EGFR mutation in exon 21. So the patient was staged and her diagnosis was T2 and not M1A disease, which is a stage 4 disease. She was a stage 4 disease because the cancer had spread to both sides of her lungs. So she began treatment with gefitinib oral gefitinib, which is a targeted tyrosine kinase inhibitor, 250 mg daily in July 2016. It was one of the very first few patients to benefit from targeted treatment as opposed to receiving the usual standard chemotherapy for advanced cancer. We followed her up with surveillance CT scan in 2017 and 2018, which indicated stable disease. However, in March 2019, she developed bronchopneumonia, which was complicated by end-stage renal failure. So this needed a temporary interruption of gefitinib, which was stopped for 3 weeks. But after 3 weeks, the patient was resumed gefitinib at the same dose, and she achieved sustained stable disease on surveillance chest X-ray. Her latest follow-up in April 2025, very recently, she remains well, indicating nearly 9 years of disease control. To highlight the remarkable points of this patient, the patient showed disease stability for more than 9 years on gefitinib. This far exceeds the median progression-free survival and overall survival from important trials such as the IPASS and NEJ002 trials, where for first-generation TKIs like gefitinib, the progression-free survival was 9 to 12 months and the overall survival was 20 to 30 months. And as you can see, these figures are much lesser than 9 years. Additionally, resistance mutations such as the T790M or MET amplification often emerge after prolonged exposure to these drugs, but this patient did not show evidence of such resistance, indicating that her tumor possessed unique genetic features that delay or prevent resistance. So there must be some unique tumor genetics in these exceptional responders, which enables them to live well and live longer than expected. So this is what has already been done in the study of these exceptional responders. Combining genomics, proteomics, and transcriptomics will allow a deeper understanding of how tumors evade resistance and adapt to treatment in exceptional responders. So this is evidenced by a few studies. One of the first studies to study this group of patients was the NCI Exceptional Responders Initiative in the US. This study revealed that distinct genomic alterations, such as DNA repair defects, were associated with unexpected durable response. Building on this, another study is by Bilosic et al., which provided further evidence that molecular profiling using techniques such as whole exome sequencing and RNA sequencing revealed 87.5% of exceptional responders in this study had non-synonymous EPHA5 mutation, and many showed changes in DNA damage repair pathways, leading to a pro-inflammatory state and effective anti-tumor immunity, possibly contributing to outlier outcomes. Interestingly, there's a new ongoing multi-center study. It's called Rosalind. It's based in the UK and it's pioneering the creation of an extensive multi-omics database of cancer outliers by molecular profiling exceptional survivors in some of the most aggressive cancer. The availability of a diverse and inclusive multi-omics database may be a standard component in future drug development to provide precision oncology worldwide. As a conclusion, the integration of molecular biology and genomics has revolutionized modern medicine by enabling the development of precision oncology, where therapies are matched to the molecular profile of an individual's tumor. And the most promising area is the study of exceptional responders. However, current molecular profiling data in exceptional responders does not include Asian populations. So this is a critical research gap which is important to address as it is both scientifically relevant and also locally impactful. In resource-constrained healthcare systems like Malaysia, this knowledge is not just academic, it has practical value because if molecular profiling can identify patients likely to achieve long benefit from all the less costly generations of drugs, it could dramatically improve not just the patient outcomes, but it also can minimize financial burden. So personalized medicine in this context becomes not just precise, but also sustainable. Thank you.